All right, this is the answer key for the digestive physiology activity. The first set of questions, so looking at the digestive tract, two events of digestion that take place in the oral cavity. So that's mastication, which is a fancy word for chewing, and saliva secretion. What digestion event happens in the esophagus? It's the esophageal transit. So it's a process we didn't get much chance to talk about. It's called peristalsis. It's basically when the smooth muscle contracts and relaxes and pushes food in a certain direction. Number 3, what digestion event happens in the stomach? This is where the food is mixed with digestive juices, the hydrochloric acid. 4, what important chemicals are released by the pancreas and the gallbladder? Bile and the pancreatic juice. And partially digestive food, reaches your small intestine and mixes with digestive juices. What is it called? This is called chyme. And then number 6, in the small intestine, what two things is this broken down into? Undigested food and then nutrients. 7, what happens during a nutrient absorption? Nutrients from the small intestine pass into the capillary beds, where the nutrients are sent to the organs. And the last question, what digestion event happens in the large intestine? So this is where dehydration or water removal happens from food. And then excretion of indigestible components of food. All right. Part 2. So we looked at chemical versus mechanical digestion. So mechanical digestion, this is any sort of physical change in food. Usually involves some muscle contraction or some grinding. And then chemical digestion involves some sort of chemical, whether that be hydrochloric acid or an enzyme. Okay, so in the mouth, chemical digestion, you have different enzymes in your saliva produced by the salivary glands. And then mechanical is mastication. Esophagus is just mechanical. It's the contraction of the food bolus, which is what the food is called at that time when it's in your esophagus, to the stomach. Stomach acid is chemical in the stomach. This is also called the gastric juice. And the mechanical, there's also a little bit of mixing of food because you have a layer of muscle, a smooth muscle that surrounds your stomach. The duodenum, this is where the bile enters your digestive system from the liver. The purpose of the bile is to emulsify fat. Emulsification, temporarily makes non-polar things polar. So fats, lipids are non-polar. They don't mix with water. But bile, which is an emulsifier, temporarily makes them soluble in water. So they can be broken down. Pancreatic juices break down macromolecules. Small intestine is chemical only. So this is where enzymes that aid in digestion come into play. And then large intestine, which is mechanical only, the peristaltic contractions move food towards the end of your rectum. Then mass peristalsis, that's a fancy word for diarrhea. That's when all of the food in all of your colons is forcefully removed. Fun fact. Okay, part 3, digestive enzymes. So I'm not going to go through these. I'll just let you pause the video here if you want to check your answers with mine. Okay, number 4, so we looked at the digestive system of a rabbit. Your answers are going to vary slightly. Essentially, the digestive system is identical. Most mammals have the same digestive process. Their digestive system anatomy is a little bit different, including the shape of the fecal matter. I don't know if you saw that on the video. Their feces are little tiny rabbit pellets, whereas human feces can vary. Okay, part 5, you looked at the intestinal absorption at the macromolecule level. So depending on what monomer you're looking at, how it gets into either the bloodstream or the lymph is a little bit different. So this is a little bit of throwback to freshman bio. We talked about different types of movement of molecules across the cell membrane. I'm not sure if you do this in AP Bio. I'm assuming you do. So glucose, so this is the monomer of polysaccharides or carbohydrates. So first enter the cell through indirect active transport. So active transport meaning it requires energy and it requires some sort of protein. Facilitated diffusion to leave or transport through the basal pole, which is the end of the cell. Facilitated diffusions that requires no energy, goes from high to low concentration, and then this passes into the blood. Amino acids and peptides, which are the monomers that make up proteins, they first get into the cell through indirect active transport, same thing as glucose, and then leave with direct active transport. So again, requires energy on the cell's part and requires some sort of protein, some sort of surface protein. And then they get into the blood. And then fatty acids and monoglycerides, which come from fats and triglycerides, the monomers, use passive single diffusion. So they pass freely from high to low concentration, no energy required, and then leave through a process called exocytosis, which is where the cell has to use energy to form a little pocket, a little vacuole around the fatty acids and monoglycerides. And then that little vacuole, or excuse me, little vesicle binds with the surface of the cell and then releases the components into the lymph. And this is called chylomicron formation. I think there was an explanation on the applet, I'm not sure. This is when fatty acids and monoglycerides are first used to form triglycerides, which are then associated with proteins, forming water-soluble macromolecules called chylomicrons. We don't need to know that. Okay, so if you have questions, let me know.